Hi everybody, welcome back to part 2 of our cell communication and stress video lectures. This is part 2. In part 1, remember we talked about mechanisms that cells use for communicating, and now we're going to talk about a specific example of cells communicating. We're going to use stress as our example. We're using stress because stress is something that everybody is familiar with. In our evolutionary past, stresses tended to be something that was acute. You would have something happen that was scary or dangerous. A predator would come by, or there would be a storm, but usually it would be over fairly quickly, and the appropriate response was the fight or the flight reaction. In other words, you either fight off or run away from whatever is bothering you. And your adrenal glands would shove off that hormone, and then when the process was over, they would relax and everything would be good until the next stress. So it was a very episodic thing. In modern times, stress is very chronic, and we all can think of lots of chronic stressors, our jobs, exams. Right now, I'm sure we're all thinking about the COVID-19 virus. These are stresses that just keep going and keep going and keep going, and your adrenal glands keep secreting these hormones. They don't get a chance to stop and recuperate, and eventually you can deplete the ability of your adrenal glands to keep secreting these hormones. Now, that can be a problem because then you can't respond appropriately if there is an acute stress that you need to respond to, but these can also lead to a number of different health problems like obesity, immune problems, heart problems. All kinds of things can happen when you're chronically stressed. We're not going to talk about those right now, but you could do a quick Google search and probably come up with 15, 20 different diseases that have a strong stress component. Now, the other type of stress that I wanted to talk a little bit about, because we know a little bit more about the genetics of these types of stress, are these acute stresses that are traumatic in nature that can lead to post-traumatic stress disorder, or PTSD. There's a number of different things that can trigger PTSD in some people. Not everybody. Health scares, natural disasters, being the victim of a crime or victim of abuse, being in combat. And the response to these traumatic events in some people is a long, ongoing thing where you've got an inappropriate response that leads to anxiety, flashbacks, avoidance of the memories, and can cause serious problems when you're trying to get through your day-to-day life. So the interesting thing about this is that researchers clearly see that people who've been exposed to the exact same traumatic experience don't necessarily respond the same way. One person might have very severe PTSD. Another person who was standing right next to them during some event might have mild PTSD, and somebody else might have absolutely no symptoms of PTSD at all. And so people wanted to learn more about this, and they've been able to do some research. And in fact, researchers looked at victims of child abuse, and amongst the many different genetic components that seem to have an effect, they were able to identify a gene called FKBP5. Let me move my arrow so you can see it. FKBP5, which had variations between different people, and those variations seem to correlate with presence and severity of PTSD. Now, this protein, FKBP5, is a protein that every cell in your body makes, and they found variations in this protein that were involved in either the genetic sequence or epigenetic changes in the genes and how it was used. And the other thing that was really interesting is that they found that glucocorticoids like cortisol affect how much of this protein is made in a dose-dependent manner. So what does that mean? Well, let's go back to the cartoon we saw before about how glucocorticoids and other steroid-based hormones work. So we saw this in the previous video. Here's our hormone. In this case, we're not going to call it testosterone. We're going to call it cortisol, which is the glucocorticoid hormone. And remember that the hormone is hydrophobic, so it diffuses into the cell, binds to a receptor. The receptor hormone complex diffuses into the nucleus, binds to specific DNA sequences, turns on mRNA, and the mRNA is used to make new protein. So in this case, cortisol turns on the gene to make FKBP5 protein. All right. And so remember I said that this is dose dependent. That means that the more cortisol that your body is secreting, the more of the FKBP5 protein your cells will make. So if you're a little bit stressed, you make a little bit of the protein. If you're severely stressed, you would make a lot of this protein. All right. Now the other thing we said is that there were genetic variations in this protein that affected how the body responded to the levels of this protein. So what does that mean? Well, there are changes in the DNA sequence, genetic variants. Another term that you might think of with this is mutations, although the word mutations tends to have the connotation of a bad change, and genetic variation is a less loaded term. It just means that there's a change in the DNA sequence. Now, this is one example shown on this slide of single nucleotide polymorphisms, or SNPs. And SNPs just mean there's one place in the gene where different people have a different nucleotide. So here we have in this figure a purple person, a blue person, and an orange person. And we're seeing the DNA for a specific gene. And you can see that for the purple person at this particular site, they have the nucleotide A. The blue person happens to have the nucleotide G, and the orange person has the nucleotide T. All the rest of the gene sequence is exactly the same. There's just one nucleotide that varies between these people. Now, single nucleotide polymorphisms don't change the reading frame of a gene. All right, so if you go back and review transcription and how the genetic code is used to make protein, you'll remember that codons are read in 3-unit nucleotide sequences. And so if you change that 3-nucleotide sequence by only one nucleotide, if it's in the first or the second position of the codon, it might change the amino acid. If it's in the third position, that wobble position, it might not change the amino acid that is inserted into a protein. In addition, these SNPs, or single nucleotide polymorphisms, might be outside of the coding region. They might be in the intron region of the gene, or they might be upstream or downstream of the gene, and so they might not affect the protein at all. So single nucleotide polymorphisms may or may not have any effect on how your genes are used and how the proteins affect your body. The other type of genetic variations are insertions and deletions, which are sometimes called indels, right? Insertion and deletion. Put those two words together. And insertions and deletions, if they happen to occur in a gene coding sequence, tend to be more serious than single nucleotide polymorphisms because they might change the reading frame of a protein. So for instance, in this example, we have our wild type sequence up at the top where we've got a sequence that's colored in either yellow or green. And the second gene sequence, a different person, there's a 3 base pair deletion. So you can see that this green section is 3 nucleotides shorter in this gene sequence than in the one above, the wild type or putatively normal gene sequence. There's basically a TAA missing. If this is a gene sequence, TAA, that's 3 nucleotides, that's 1 codon's worth. You might change 1 or 2 of the amino acids, but it's not going to change the entire, well, it might change. It may or may not change the coding sequence depending on if you're deleting an entire codon or parts of 2 codons. That could change everything downstream. The other sequence at the bottom here is showing you a 4 base pair insertion. You can see in orange there's some sequences that were added, and that's definitely going to change the reading frame of every amino acid downstream of that insertion. So you can see how changing the insertion, the reading frames, changing the amino acids that are put into a protein, or maybe adding a stop codon much sooner than it should have been, can have severe effects on how a protein is made and how it functions. All right, now the other thing that FKBP5 protein changes that had been noticed were some epigenetic changes. And so I just wanted to remind you that epigenetic changes are the changes in how genes are used based on whether the chromatin, the DNA, is loose or tightly packed. And I'm going to try and show you a YouTube video. If it doesn't work, I will stop and I will just tell you to go and look at this URL, which is also in Canvas, talking about epigenetic changes. And I will just also let you know, in case it doesn't work and I have to cut out the end of this video, that the terms and concepts to know for this portion of the lecture are present as well. So let's try that YouTube video. Here it is. It's only 3 minutes long.
The sweet smell of fruit doesn't normally send rats running, but when researchers paired the orange cherry almondy scent of the chemical acetophenone with a painful electric shock, lab rats quickly learned to fear it. Along the way, extra neurons sprouted in their noses and in the smell processing center of their brains, making them super sensitive to the scent. This result isn't shocking. What is surprising is that the rats' pups and their pups' pups were also startled by the smell of acetophenone and had the same extra neurons as their fathers, despite never having been introduced to either their dads or the fruity scent before. But how could the pups have inherited something that their fathers learned? Basic genetics tells us that only DNA gets passed along to offspring. Characteristics like memories, scars, or giant muscles can't get passed on since acquiring them doesn't alter the genetic code. But it turns out that instilling fear in the rats did trigger genetic changes, not in the DNA sequence itself, but instead in how that code was read and used in the rats' bodies. In every cell, biological machinery constantly translates DNA into the proteins needed to carry out vital processes. Chemical switches attached to the DNA turn genes on or off or up and down, telling the machinery which proteins to produce and in what quantities. These switches, called epigenetic tags, are why a kidney cell looks and acts differently than a skin or nerve cell, even though all 3 cells have identical DNA. But the switches in any one cell aren't set in stone. Teaching those rats to fear the fruity smell switched one of their smell sensing genes into overdrive. Researchers don't know all the places in the rats' bodies where this switch got flipped, but they know it happened in one key set of cells, the rats' sperm cells, which would one day pass along the tweaked genetic material, making the next generation of rats super sensitive to acetophenone. Rodents aren't the only creatures demonstrating this weird type of inheritance. In Överkalix, Sweden, boys who suffered through tough winter famines went on to have super healthy sons, with extremely low rates of heart disease and diabetes. And their sons had the same excellent health, living an unbelievable 32 years longer on average than the grandsons of boys who hadn't gone hungry. To be clear, this does not mean that we should start starving our kids for the benefit of future generations. Scientists don't even know yet exactly which switches the Swedish famines flipped. While we have been able to connect specific epigenetic changes to health effects in mice, we're a long way off from being able to make those connections in humans. That may sound like a bummer, but it's mostly because we humans don't live in the well-controlled environment of a laboratory. And for that, we should be grateful.
Okay. That is that. Thank you very much for looking at this. I will be working on adding some questions for you to answer so that we can get some points associated with these lectures, and more lectures will be coming in the future. Hopefully things will get smoother and smoother as we go along. Take care and stay healthy. Bye-bye.